Hello everyone, welcome to today's live broadcast, 1.25 dihydroxyvitamin D testing and its clinical significance. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Diasorin. Diasorin Incorporated offers the broadest, fully automated infectious disease and specialty diagnostic menu, including the best-in-class vitamin D. These quality assays are analyzed on the versatile Liaison XL platform. They provide value through sales, service, and support solutions to clinical laboratories by consolidating their menu in order to streamline processes and increase efficiency. For more information, please visit diasorin.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or to submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I'd like to introduce today's speaker, Dr. Armin Zittermann. Professor Dr. Armin Zittermann is the head of the Center for Clinical Studies in Thoracic and Cardiovascular Surgery at the Heart and Diabetes Center in Germany. Dr. Zittermann also lectures at the Institute of Nutrition and Food Sciences at the University of Bonn on specific aspects of food security. His research focuses include the prevalence and consequences of insufficient levels of vitamin D in humans, in particularly in patients with cardiovascular diseases. Furthermore, Dr. Zittermann investigates the confounding factors on outcomes of cardiac surgery patients, including the influence of immunosuppressants in heart transplant patients. His scientific work includes the various fields such as biochemistry, physiology, epidemiology, nutrition, heart surgery, and statistics. Dr. Zittermann has published more than 200 journal and book articles and has presented more than 250 lectures, national and international. He is a reviewer for a number of international scientific journals and serves on the editorial board of various nutritional medicine journals. I'll now turn it over to Dr. Zittermann for his presentation.
Hello everyone, also from me. First I will give you an overview and will make some general remarks on vitamin D. Then I will talk about laboratory aspects, about actions of vitamin D, about clinical aspects, and then I will draw some conclusions. The group of steroid hormones consists of 4 members. And vitamin D is one of these 4 members. And as you probably know, there are 2 vitamin D sources for humans. Vitamin D synthesis in the skin after UVB exposure and oral vitamin D intake. And vitamin D itself is inactive and it has to be metabolized in the human body, first in the liver, into 25 hydroxyvitamin D, and if the vitamin D or the 25 OHD level is low in the human body, you can prevent this by vitamin D supplements. However, the active hormonal form of vitamin D is the 1,25 dihydroxyvitamin D, and it is produced in the kidneys. And it is tightly regulated. And 2 minerals are very important for the regulation of 1,25 dihydroxyvitamin D, and this is plasma calcium and plasma phosphate. And this regulation is mediated by 2 hormones. the calciotropic PTH is increased. For example, if plasma calcium levels are low, and then PTH is responsible for an increase in circulating 125 dihydroxyvitamin D levels. And the other hormone is FGF23. It's a phosphaturic hormone, and this hormone is upregulated if plasma phosphate levels are high. And this upregulation of FGF23 results in a decrease in circulating 125 dihydroxyvitamin D levels. And now I will come to some laboratory aspects. Up to now, 125 dihydroxyvitamin D measurement has been neglected for several reasons. First, there is a homeostatic regulation in circulating 125 dihydroxyvitamin D levels, at least in healthy individuals. Second, there is the active hormonal form of vitamin D has a relatively short plasma half-life. And the third aspect is that up to now, the measurement was very difficult, and high sample volumes have been required at extensive purification and separation. And here you can see data from a meta-analysis. And these data, they show the results of studies of vitamin D supplements on circulating 125 dihydroxyvitamin D levels. And here you can see that the RIA test kits, they show different results compared to other test kits. And this demonstrates that up to now, the measurement of 125 dihydroxyvitamin D was difficult. And if we look at the actions of 125 dihydroxyvitamin D, we can say that almost all tissues in the human body have 125 dihydroxyvitamin D receptors. For example, muscle, bone, heart and circulation, brain and nervous system, and also the immune system. And the active form of vitamin D has genomic and non-genomic cellular vitamin D actions. And therefore, it's a classical steroid hormone, and the genomic actions are mediated by a cytosolic vitamin D receptor. The 125 dihydroxyvitamin D in the circulation, it binds to this cytosolic vitamin D receptor. And the 125 dihydroxyvitamin D can also bind to a membrane-bound vitamin D receptor. And this receptor is responsible for the non-genomic vitamin D actions. But it's also possible that 25 hydroxyvitamin D is uptaken by some tissues, and they are able to produce the active hormonal form of vitamin D by themselves. And here you can see that several types of tissues are able to produce 125 dihydroxyvitamin D by themselves. For example, immune cells, also bone cells, cells of the gastrointestinal system, also cells of the vascular system, and for example, pancreatic beta cells. And we know that vitamin D has several modes of action. It's well known that vitamin D has endocrine functions. As I mentioned before, it's important for the regulation of two minerals, of calcium and phosphate in the plasma. But it has also autocrine and paracrine vitamin D actions in several tissues. For example, in the immune system, also in the beta cells of the pancreas, and it's also important in tumor cells for the regulation and differentiation of these cells. And it's important to know that the uptake of 25-hydroxyvitamin D into the target cells, where the 125-dihydroxyvitamin D can be made by these cells themselves, that this uptake depends on the concentration of 125-dihydroxyvitamin D in the circulation. For example, here you have patients with renal impairment, and if these patients they have a lower uptake of 25-hydroxyvitamin D into the target cells compared with patients who have normal kidney function. And if you administer the active hormonal form of vitamin D, 125-dihydroxyvitamin D, which is also called calcitriol, to these patients, the uptake of 25-hydroxyvitamin D into the target cells is also increased. And now I want to come to some clinical aspects. 125-dihydroxyvitamin D can be measured in the circulation, and the reference range is between 25 and 87 picogram per ml. And it's well known that the circulating level of 125-dihydroxyvitamin D is low, for example, in patients with chronic kidney disease. It's also low in patients with hyperparathyroidism or pseudo-hyperparathyroidism, and it's high in patients with primary hyperparathyroidism. And in these cases, one can use measurement of 125-dihydroxyvitamin D for confirmatory reasons. But we can also use it for diagnostic reasons. For example, some patients may have hypercalcemia, and this hypercalcemia may be induced by high 125-dihydroxyvitamin D levels in the circulation, for example, in patients with sarcoidosis or with tuberculosis. And here we can use the test for measurement 125-dihydroxyvitamin D. And there are also two forms of vitamin D-dependent rickets. Type 1, where the 1-alpha-hydroxylase is very low, and the circulating 125-dihydroxyvitamin D is very low. And also vitamin D-dependent rickets type 2, where the 125-dihydroxyvitamin D is very high in the circulation because there is a vitamin D receptor defect. But these types of vitamin D-dependent rickets are very rarely seen in the clinical setting, and they are not the same as the nutritional vitamin D rickets. But it's important to use the test to differentiate these different forms of vitamin D-dependent rickets. And now I want to talk about some diseases. First, about chronic kidney disease. And it's well known that in aging Western societies, chronic kidney disease is very prevalent. And we also know that in patients with renal impairment, also the circulating 125-dihydroxyvitamin D level goes down. And for example, in patients with CKD stage 3, we have usually a level below the reference, the lower limit of the reference range. This means between below 25 picograms per ml. And here you can see that if plasma 125-dihydroxyvitamin D levels decrease, then there is an increase in the risk of death. Or in other words, if there is an increase per 10 picograms per milliliter in plasma 125-dihydroxyvitamin D, then this is associated with an with a better survival in the patients. And this is also independent of age, ethnicity, diabetes, cardiovascular disease history, PTH levels. The only factor which was attenuated this aspect, this association between 125 dihydroxyvitamin D levels and poor clinical outcome, was FGF23. And this is understandable because this is a very important hormone which regulates the circulating level of 125 dihydroxyvitamin D. And this study could also show here, if you look at those patients who have very low 125 dihydroxyvitamin D levels, for example, below 15 picograms per milliliter, this was associated with a higher risk of death or progression in chronic dialysis, and compared to patients who had 125 dihydroxyvitamin D levels above 25 picograms per milliliter. And in the right-hand column, you can see that those with 125 dihydroxyvitamin D levels between 15 and 22 picograms per milliliter, they had a risk of death or progression of chronic dialysis in the mediate range. So the 125 dihydroxyvitamin D is a good predictor for poor clinical outcome in patients with chronic kidney disease. And here you can see a meta-analysis about the hazard ratio of all-cause mortality for vitamin D treatment versus no treatment in chronic kidney disease patients. And you can see, if you look at the summary on the bottom line, then you can see those who were treated with vitamin D, with the different forms of vitamin D, with the active form of vitamin D and also with the native form of vitamin D, they had a lower risk of all-cause mortality, approximately 25% lower risk of all-cause mortality. And another meta-analysis on activated vitamin D in all-cause mortality could show that that there was an effect over a time period of 3 years and also of 5 years, and the risk ratio was reduced by approximately 1/3. But we also have to keep in mind that these are observational studies and meta-analysis of observational studies, and that we still need more randomized controlled trials to prove these associations and to prove causality. And here you can see the effect of activated vitamin D on circulating 125 OHD levels in the lower part of this slide. And what you can also see that the effect of activated vitamin D on circulating 125 OHD is only based on 3 studies. This means that only 3 randomized controlled trials are available, and that we need more randomized controlled trials to look on the effect of activated vitamin D on circulating 125. And you can also see that there was a wide range in the effect of activated vitamin D on the circulating level. And therefore, I personally believe that it's very important to do the measurement of 125 dihydroxyvitamin D also in patients who are treated with activated vitamin D, and to do follow-up measurements of 125 dihydroxyvitamin D levels in order to see whether this treatment is able to result in a circulating 125 dihydroxyvitamin D level, which is in the reference range. And now I want to talk about some lifestyle aspects. And here you can see that, for example, during the summer season, the 25 hydroxyvitamin D levels are higher in healthy individuals than during the winter season because of the UV exposure and the skin synthesis of vitamin D. And this higher 25 hydroxyvitamin D level is also associated with a higher 125 dihydroxyvitamin D level in the circulation. Therefore, it's important to guarantee adequate vitamin D status, and this has also an effect on the circulating 125 dihydroxyvitamin D level. But not only the vitamin D supply is important, this investigation could show that also other diseases are obviously able to influence the circulating 125 dihydroxyvitamin D levels. Here you can see that, for example, the kidney function, GFR, and also renal replacement therapy has an independent and significant aspect influence on the circulating 125 dihydroxyvitamin D, as already been shown. But in this slide, you can also see that, for example, diabetes or diuretics or the CRP level is inversely related, significantly inversely related to circulating 125 dihydroxyvitamin D levels. And approximately 20 years ago, I had the opportunity to do a study during the German-Russian space mission in a German cosmonaut. And what we did, we measured the PTH, the 25 hydroxyvitamin D, and also the 125 dihydroxyvitamin D pre-flight, in-flight, and post-flight. And what you can see here is that 25 hydroxyvitamin D and also the PTH remained fairly constant during this time. However, there was a marked decrease in 125 dihydroxyvitamin D levels until the end of the flight, and also during the first days post-flight. This demonstrates that the 125 dihydroxyvitamin D level is not tightly regulated, and there are situations where there are marked influences and marked fluctuations in the circulating level of this hormone. And as we all know, physical activity is very important for a healthy life. And we should do physical activity every day. And there are several reasons why physical activity is good for our health. And interestingly, physical activity has also a positive effect on circulating 125 dihydroxyvitamin D levels, as seen in this slide. For example, physically active individuals in the left-hand side of this slide have higher circulating 125 dihydroxyvitamin D levels than young sedentary men. And also immobilization has an effect on the circulating 125 dihydroxyvitamin D level. As you can see on the right-hand side, immobilization is associated with a decrease in 125 vitamin D, and remobilization is associated with an increase in the 125 dihydroxyvitamin D. And now I want to talk about another disease. It's heart failure. And similar to chronic kidney disease, heart failure is very prevalent in the aging Western society. And heart failure patients have several derangements in the metabolism. And one of these derangements are imbalances in calcium and phosphate metabolism, and this can also lead to advanced heart failure. And it's well known that the FGF23, the phosphate-regulating hormone, which is important for the regulation of circulating 125 OHD, is increased in patients with heart failure, and it's a very good predictor of high levels are a very good predictor of poor clinical outcome in these patients. And not only the FGF23, but also the 125 dihydroxyvitamin D is a good predictor of clinical outcome. Here we have the results of a cohort study in 500 patients with a high prevalence of end-stage heart failure. And you can see the one-year mortality was significantly higher in those patients who had baseline 125 hydroxyvitamin D levels below 25 picograms per milliliter. And this is another study from a group from Belgium, and they could show similar results. They could demonstrate that in patients with heart failure, cardiovascular death was significantly higher in those patients who had low initial 125 dihydroxyvitamin D levels. And what we also did, we looked at the outcome in patients with end-stage heart failure and on clinical outcomes, for example, death, heart transplantation, and mechanical circulatory support implants. And we were able to use a risk score, and this risk score was consisted of 5 different biochemical parameters. And one of these parameters was 125 dihydroxyvitamin D. Low levels of 125 dihydroxyvitamin D was associated with a higher risk of poor clinical outcome, and also high levels of FGF23 was associated with a higher risk. And it was possible to reach 5 points with this risk score, and those who scored 5 points, they had a much higher risk of poor clinical outcome than those with 0 points with this risk score. And what we also investigated was the association between mechanical circulatory support implants and circulating 125 dihydroxyvitamin D levels. At the beginning, just before implantation, all patients had very low 125 dihydroxyvitamin D levels. But interestingly, the non-survivors, those who did not survive the first post-operative year, they had the 125 dihydroxyvitamin D level remained very low, but in those who survived the first post-operative year, in those patients, the 125 dihydroxyvitamin D level increased significantly until the end of the follow-up period. This also shows that obviously the 125 dihydroxyvitamin D is a good predictor of poor clinical outcome in heart failure patients. And here you can see that this association is also present in heart transplant recipients. Again, you can see that those who had the lowest 125 dihydroxyvitamin D levels at the end of the 1 year follow-up period, at the beginning of the 1 year follow-up period, they had the lowest survival, and those with the highest 125 dihydroxyvitamin D level just after heart transplantation, they had the highest survival at the end of the first post-operative year. And these were multivariable adjusted data. All these data I showed you are multivariable adjusted, and they indicate an independent association between the circulating level of 125 dihydroxyvitamin D and clinical outcome. And therefore, to my opinion, we can add another aspect. The measurement of 125 dihydroxyvitamin D can also be a risk marker of premature death in heart failure patients. And there is some evidence that probably 125 dihydroxyvitamin D is also predictive for survival in other groups of patients. We need more research in this field, but it's interesting, for example, that in patients with sepsis, that those who did not, who had very low 125 dihydroxyvitamin D levels initially, that they had a relatively high mortality rate, and that in a multivariable model, 125 dihydroxyvitamin D, total calcium and age were very important predictors of 30-day survival in this study. Nevertheless, there are several gaps in present knowledge about the circulating 125 dihydroxyvitamin D level. As I mentioned before, only a few randomized controlled trials have investigated the effect of activated or of native vitamin D on circulating 125 OHD levels. And it's also unclear whether there is causality between low circulating 125 dihydroxyvitamin D and poor clinical outcome. Several observational studies indicate that there may be an association, but we need more randomized controlled clinical trials. And also the effect of different diseases and of some lifestyle factors like physical activity on circulating 125 dihydroxyvitamin D has to be studied more detailed. So now I can come to my conclusion. Circulating 125 dihydroxyvitamin D is less tightly regulated as being assumed so far, and may in future become a reliable predictor of clinical outcome in some clinical conditions. More attention should be paid on follow-up measurements of circulating 125 OHD in patients treated with activated vitamin D. However, it's also necessary to look at the plasma or serum calcium level and urinary calcium, because one activated vitamin D can also lead to hypercalcemia, and this has to be prevented. Therefore, it's important to measure these calcium levels, but also it's important to do follow-up measurements of the circulating 125 dihydroxyvitamin D to prevent high levels of this active hormone in the circulation. Thank you for your attention.
Well, thank you for that informative presentation. Before we get started on a question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can as time allows. So, Doctor, our first question is, would you explain the ways to prevent low levels of active vitamin D?
Yeah, there are several aspects which are important. First, it's important to have an adequate vitamin D intake. And for example, if you do not have adequate UVB exposure to your skin, you can use vitamin D supplements. But it seems to be also that it's important to do adequate physical activity, because physical activity can increase 125 dihydroxyvitamin D levels. And in this context, it's also important to prevent immobilization. And there seems to be an association between dietary phosphate intake and 125 dihydroxyvitamin D. Diets high in dietary phosphate, they may suppress the active hormonal form of vitamin D. So you can, even healthy individuals can do a lot to prevent low circulating 125 dihydroxyvitamin D levels.
Thank you. Our next question is, what is the definition of at risk for chronic kidney disease?
Yeah, those who have a chronic kidney disease stage 3, 4, or 5, they are at risk for low circulating 125 dihydroxyvitamin D levels. And those who have CKD stage 3, they may use a vitamin D supplement. However, those who have CKD stage 4 and 5, they should use activated vitamin D.
And our next question is, what causes the 1.25 dihydroxyvitamin D decrease in spaceflight, so in low gravity conditions, also in sedentary lifestyles?
Yeah, it's a very good question. One aspect may be the calcium increase in the circulation because of bone loss. For example, during microgravity, there is a bone loss. It's well documented, and this leads to an increase in plasma calcium. And this plasma calcium level is responsible for downregulation of the circulating 125 dihydroxyvitamin D. But this is probably not the whole story. For example, there is accumulating evidence that inflammatory cytokines suppress the circulating 125 dihydroxyvitamin D level. And for example, during the German-Russian space flight, there was a lot of stress during this mission because there was a fire on board the station. And it's known that inflammatory cytokines are increased in case of stress. And this may also influence the circulating 125 dihydroxyvitamin D level in these conditions.
Well, it looks like with the time that we have allotted, we have time for one more question. How do you explain the 1.25 dihydroxyvitamin D suppression in heart failure patients?
Yeah, these patients have very high FGF23 levels. And as I mentioned before, the FGF23 is a very good predictor for poor clinical outcome in heart failure. And the FGF23 is a phosphate-regulating hormone, and it's upregulated in case of high plasma phosphate levels. And this hormone is also responsible for the downregulation of circulating 125 dihydroxyvitamin D levels.
Well, Dr. Zittermann, I want to thank you very much for your time, not only for your wonderful presentation, but also for going through the questions that we have today. And before we let you go, do you have any final comments for today?
Yes. The active hormonal form of vitamin D is a very potent steroid type hormone, and there is accumulating evidence that it's influenced by lifestyle factors, but also by several diseases. And therefore, to my opinion, we should pay more attention to the circulating 125 dihydroxyvitamin D level in the clinical setting, but also we have to do more scientific research in this field.
Well, thank you again, Doctor, for that wonderful presentation. I would also like to thank our sponsor, Soren, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May 2017. You'll receive an email alert from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to any colleagues who may have missed today's live event. Until next time, goodbye.